Okay, today I will be talking about starch. Starch is widely distributed in plants as a storage carbohydrate, and it provides 70 to 80% of the calories worldwide. It's a polymer of D-glucose and occurs as small granules, and it's often stored in the plant seeds. It's a mixture of two glucans, amylose and amylopectin. In addition, they can also contain lipids and proteins. Amylose is composed of 20 to 30% of a normal starch molecule, and it's linear and it is a polymer of alpha D-glucose. It contains alpha 1,4-glycosidic linkages, has a very small molecular weight, and it occurs as a helical shape. One turn corresponds to 6 glucose units, and hydrogen bonds act to stabilize it. It also has a water-filled central channel, and it can form an inclusion with iodine where it forms a blue color. Amylopectin is composed of 70 to 80% of a normal starch molecule, and it's a highly branched and very large molecule. It contains alpha 1,6 linkages, which form the branching part, but the core is also composed of alpha 1,4-glycosidic linkages. The branches are relatively short and usually have 15 to 30 glucose units, and they occur as double helixes. Amylopectin can be degraded by beta amylase up to the branching points, and then what's left is known as the limit dextrin. Amylopectin can also form a complex with iodine, but it forms the red color instead. Granule structure, it can range in size from 2 to 100 micrometers, and in the native form it has a Maltese cross, which is the radial arrangement of the starch molecules, and it's a crystalline structure. The center is called the nucleus, and it grows out from the nucleus. And once gelatination takes place, as we'll see later, the Maltese cross is lost. The granule is usually 70% amorphous and 30% crystalline. Crystalline is a very compact, dense structure, whereas amorphous is like loose and less compact. Amorphous is mainly composed of amylose, whereas amylopectin is the major contributor to crystallinity. And this was determined because although amylopectin is branched, amylose can be dissolved out of the granule, and the crystalline structure still remains. And this was determined through X-ray diffraction. Crystallinity occurs due to the alignment of amylopectin branches or the double helixes between amylose, and they're stabilized due to hydrogen bonding. Granules are completely insoluble in cold water, but upon heating they start to swell at the temperature that's known as the gelatination temperature. And they, as we'll see, they lose crystallinity during this process. So gelatinization, which is this process, to put briefly, it's granule swelling, exudate release, which is largely amylose that comes out, and granule disruption. Okay, so gelatinization, the process is granules are suspended in cold water, and the starch granules start to swell with an increase in diameter up to 30 to 40%. If it's heated, irreversible changes take place, and this takes place at what's known as the gelatinization temperature. This is usually around 50 to 70 degrees Celsius, but it depends on the type of starch that we're talking about. So the granule absorbs 20 to 40 grams of water, and the viscosity rises steeply. As you can see, this is a measure of temperature versus viscosity, and it absorbs water. The viscosity rises steeply to this point, where it's the max viscosity. And then as the amylose diffuses out of the cell, which is what you can see in the fragments, they go into the solution. And finally, the granule bursts, so it's down here where you just have amylose in the solution. In more detail, the starch granules melt when they're heated in the water, and the polymer network that's formed of hydrogen bridges are disrupted. So the amorphous or irreversible swelling, which is what you can see happening in this portion, causes the dissolving out of amylose. So the continued heating results in the granule swelling and the additional leaching of amylose. And eventually this causes disruption, and it forms what's called a paste. And then from this point on, the cooling of the paste results in a viscoelastic gel. And we'll talk about that later, the difference between a gel and a waxy solution. And just to note again, the viscosity increases to a maximum when the gel swells due to the heating. Okay, so differences between amylose and amylopectin. Amylose, if starch is high in amylose, it's usually known as a gel, and it's capable of doing retrogradation. But amylopectin, starches that are high in amylopectin, are waxy starches and do not undergo retrogradation. Waxy starches, again, are non-gel formers and do not retrograde, and they're high in amylopectin, whereas non-waxy starches are high in amylose and they form gels and have hydrogen bonding. Okay, retrogradation is the irreversible transition from the solubilized or highly swollen state to an insoluble or microcrystalline state. Hydrolysis, in one way to reduce this is to eliminate it to around 20 to 30 chains, and it has a low chance of doing retrogradation. And this can be applied to bread. Freezing bread is actually more effective than putting bread in a fridge. The fridge temperatures actually accelerate retrogradation, whereas freezing acts to prevent retrogradation. So just to look at this, this is the unswollen and more of a crystalline structure, and then as a swollen, the hydrogen bonds are broken, and you get the amylose in the solution. And if you have a slow process, it happens retrogradation, and you have a precipitate form, which is insoluble and it has a very tight structure. Whereas if you have rapid cooling, you form a gel.